My name is Matthew Pickering, and this lecture is on the complement system. Complement was first recognized as a heat-label component within serum that augmented the ability of antibodies to destroy bacteria. The key effector functions are opsonization, leukocyte activation, and cell lysis by the membrane attack complex, which produces pores in cell membranes. It is a complex network of proteins that consist of three activation pathways. It is important to consider the triggers of these pathways, how they are regulated, and how they contribute to the effector functions. I first summarize the functions of complement. Complement is a key component of host defense, and it contributes to the killing of pathogens through opsonization, leukocyte activation, and membrane damage by the membrane attack complex. Complement lowers the threshold for an antibody response to antigen, and complement has been referred to as a natural adjuvant. C3d is a molecular adjuvant of innate immunity. The CD19-CD21 complex is an essential B-cell co-receptor that functions synergistically to enhance signaling through the B-cell antigen receptor in response to T-cell dependent complement-coated antigens. Complement has a key role in immune complex processing, that is, the removal of antibody-antigen complexes. Immune complexes coated with C3b, that is opsonized, interact with complement receptor 1 on red cells, and this enables the removal of the complexes by phagocytes within the liver and spleen. There are many unexpected roles for complement, for example, a role for complement C1q in the physiological removal of apoptotic cells. The interaction between C3 and CD46 influences the CD4 T cell Th1 response. C1q has been shown to act as an angiogenic factor, and there is growing evidence for a role for intracellular complement in promoting differentiation of T helper cells into Th1 cells and influencing cellular metabolism. This schematic summarizes complement activation. The classical pathway is triggered by immune complexes, that is IgG or IgM complex with antigen. These complexes will interact with C1q, which is the first component of the classical pathway. The pathway activation results in the production of enzyme complexes that can proteolytically cleave complement C3. These enzymatic complexes are termed C3 convertases, and they result in the enzymatic cleavage of C3 to C3b. The classical pathway C3 convertase is composed of activated complements C4 and C2, which is denoted C4b2a. The lectin pathway is triggered by the interaction of mannose-binding lectin with carbohydrate structures, analogous to the interaction of C1q with immune complexes. And it can also be activated by the interaction of phycolins with acetylated structures. The lectin pathway shares activation proteins with a classical pathway such that it results in the production of an identical C3 convertase denoted C4b2a. In contrast to the classical and lectin pathways, the alternative pathway is constitutively active due to an unstable internal thioester bond in C3, which is susceptible to hydrolysis. This results in a C3 molecule that can interact with factor B. Following enzymatic cleavage of factor B by factor D, the alternative pathway C3 convertase is formed. This is denoted C3b-BB. Because the pathway is constitutively active, it requires constant regulation to prevent uncontrolled activation of C3. So activation of all three pathways results in the generation of C3 convertases. These cleave C3, generating fragments termed C3a, an anaphylatoxin, and C3b, an opsonin. Rapid amplification of C3b is achieved through a positive feedback loop, termed the C3b amplification loop. This can generate millions of C3b molecules within minutes. Through the binding of an additional C3b molecule, C3 convertases become capable of cleaving complement C5, and these are termed C5 convertases. These generate C5a, an anaphylatoxin, and C5b, which, through the sequential interaction with complement C6, C7, C8, and multiple molecules of C9, generates the membrane attack complex C5b9. It is important to note that C3b can be degraded proteolytically into other components called IC3b and C3DG. Unlike C3b, neither IC3b nor C3DG can contribute to further C3b generation, so these steps switch C3 activation off. However, these components mediate biological functions through the interaction with cell surface receptors, and these are C3b binds to CR1 or CD35 on red cells, mediating immune complex transport, and also monocyte macrophages mediating phagocytosis. IC3b binds to CR3, CD18-11b on monocyte and macrophages mediating phagocytosis, and C3d binds to CR2 or CD21 on B cells and follicular dendritic cells, influencing the antigen response. Note also that the anaphylatoxins C3a and C5a mediate their functions through receptor binding. Regulation of complement is tightly controlled by a complex group of proteins that act at different steps in the pathways. The key regulators are indicated in red on the schematic, and which can act in plasma or on cell surfaces or both. Complement regulation is critical to limit damage to host tissues and to prevent depletion of proteins. Regulatory proteins act by preventing assembly of C3 and C5 convertases. For example, factor H prevents the assembly of the alternative pathway convertase. Accelerating the natural decay of the convertases, for example, the appropriately named cell surface protein decay accelerating factor or CD55, promotes dissociation of the convertases. Degradation of C3b to IC3b by the enzyme factor I with cofactors such as factor H prevents further C3b amplification. The membrane regulatory protein CD59 prevents the assembly of the membrane attack complex, and factors I and H have a critical role in regulating both the alternative pathway and the C3b amplification loop. We can learn a lot about the function of complement by studying individuals with either activation protein deficiency, too little complement activation, and those with regulator protein deficiency, too much complement activation. Activation protein deficiency is associated with susceptibility to bacterial infection, and there is a strong association between terminal pathway deficiency, so deficiency of C5, C6, C7, C8, or C9, and susceptibility to Neisseria infection. Deficiency of C1q is associated with the development of systemic lupus erythematosus, and this may relate to the role of C1q in the clearance of apoptotic cells. Regulatory protein deficiency includes C1 inhibitor deficiency and angioedema, paroxysmal nocturnal hemoglobinuria or PNH, where the genetic defect results in red cells lacking GPI-anchored proteins, which include CD55 and CD59. The lack of CD59 results in red cells that are susceptible to lysis by the membrane attack complex formation, which is triggered through alternative pathway. And there is a strong association between abnormalities in the alternative pathway, for example, defects in factor H, and complement-mediated kidney diseases. These include a glomerular disorder driven by abnormal C3 deposition, termed C3 glomerulopathy, and a glomerular disorder driven by C5-dependent thrombosis, termed atypical hemolytic uremic syndrome. Note that these are rare deficiency states which are driven by mutations and which result in extreme complement dysregulation. Importantly, we now know that ostensibly subtle variation in complement regulation, which is mediated by polymorphisms within complement regulator genes, influences susceptibility to common disease. Examples include the strong association between susceptibility to age-related macular degeneration, the commonest cause of acquired blindness in the developed world, and polymorphisms within complement proteins. And similarly, polymorphic variation in the complement factor H protein family influences outcome in IgA nephropathy, the commonest glomerulonephritis. Complement therapy is a growing area, and examples include the prevention of C5 activation by monoclonal antibodies, eculizumab, ravulizumab, which is used to prevent glomerular thrombosis in atypical hemolytic uremic syndrome, and to prevent red cell lysis in paroxysmal nocturnal hemoglobinuria. Remember, I highlighted the importance of the terminal pathway in visceral infection. One of the predictable side effects of anti-C5 therapy would be a risk of visceral infection, meningitis, and this is what can happen. Consequently, the risk of visceral infection for individuals on anti-C5 therapy needs to be mitigated, and this is done through vaccination and antibiotic prophylaxis. Other agents include a C3 inhibitor used in PNH and a C5a receptor blocker used in anti-neutrophil cytoplasmic antibody-associated vasculitis. For the commonest activation protein deficiency state, C1 inhibitor deficiency, treatment is with C1 inhibitor concentrates. In summary, I have discussed complement function, the pathways emphasizing their triggers and regulation, complement and disease, and the therapeutic inhibition of complement activation.